concerned about, right, especially nowadays, are chemicals from the environment. And this is probably the part of this chapter where the author really, really allows himself to wallow in biochemical details. There are lots of chemicals that that cause mutations, and you'll see in the thing. So some of them get included because they are pretty much the same shape as a nitrogenous base, and the enzymic systems don't discriminate against them. Some of them change existing nucleotides. They might add alkyl groups, or they might deaminate one of the nucleotides. They might be oxidizing agents. And some of them slip in between the stacked base pairs in the interior of the thing. Those things are called intercalating agents. One of those is pretty infamous. It used to be in our food. It was a dye that people used to make food more attractive, but it turns out it's mutagenic because it's sort of a flat, planar molecule, and it slips into the double helix and causes problems. But there's a range of these, range of chemicals. There's only, I think, one important thing to remember from this chunk of the chapter. The rest of it's all chemistry and not genetics. So, let me take one example. One of these base analogs is a chemical called 5-bromouracil. And it's a base analog of thymine. With me so far? So, if, for example, we had a little piece of DNA sequence, let's say that was the sequence, and it had been replicated, and now, instead of putting a thymine there, a thymine-containing nucleotide, it is, the base is 5-bromouracil. With me so far? So, because this thing chemically acts like a regular old base, it has been incorporated into a nucleotide and stuck there. Now, 5-bromouracil will do complementary base pairing with G. It's not right, right? It's a substitute for a T, which would pair with A. But it does complementary base pairing with G. So here's the way to think about this. Here is my mutation. Right, it happens, a chemical mutation. When we replicate the DNA, this strand we can kind of forget about because it's normal. Right? It's this strand, when it gets replicated, that is interesting. Here's the single strand. We've separated our strands. We're doing the semi-conservative, and we've built the complement here. But now it goes to be a G. And if we have a further round of replication, it won't even be recognizable, right? It's not like this strand has any recognizable problem. This strand still will, and maybe that can be repaired. Maybe this can be repaired, but this strand, that original chemical change is completely unrecognizable. So what has happened here is that an AT base pair has gone to a GC, right? The only thing that I think is valuable in this whole huge chunk of the chapter is being able to think about, if there's this error, what do you end up with? If there is this particular error of an incorporation of 5-bromouracil, what you get is a change from what was originally an AT to GC. This obviously is a change from one purine to the other one, right? A to G. Yes? The two purine bases, A and G. We've gone from one purine to another, and that gets called a transition. If it goes from purine to pyrimidine, or vice versa, it's called a transversion. It's one of the curiosities of mutation that there are more ways you can go from purine to pyrimidine or pyrimidine to purine. There's just more options, right? You could go from an A to a C or a T instead of just going A to G. Even those transversions, there's more different ways you can have a transversion. They're rarer in mutation for some particular reason. Transitions tend to be the common one, the common type of these substitutions. Okay, so what I think is valuable about this, right, is that mutation seems scary and unpredictable and mysterious, and just chemically, you can get this information for any mutagen. What does it do? It causes this. It's a base analog. It's an alkylating agent. Once it's has alkylated an adenosine residue, adenosine mispairs with whatever. And then you can think, okay, I can just trace this through. I can see what kind of mutation will arise in this situation. So there's chemicals. What else is there from the environment that causes mutation? What else causes mutation that just comes from the environment? Yeah, ionizing radiation, including sunlight.
Isn't how that lady died from radiation cancer or something like that? Oh,
So
people don't want to see
the sunlight's a really an interesting one, right, for us here in Florida. And it has a sort of a rather peculiar effect, right? So for UV, if we can think about our sugar phosphate backbones,
what is this, poly?
this is a piece of DNA that contains two thymine residues in a row.
No, but like the Muslims and all of that, I think they didn't bug. Just
what UV does is it creates a thymine dimer. It connects these carbons like this, so you get a 4-carbon ring. And this is incredibly difficult for replication. It's a huge mechanical problem when the replication machinery comes to it. And so the efforts to overcome that thymine dimer, or the pyrimidine dimer, creates mutations of their own. So this is our homegrown one. The sort of nuclear radiation, it tends to cause double strand breaks. And the effect of whether that causes mutation or not depends a lot on how that break gets repaired. A few weeks ago, I was talking about the molecular basis of recombination, right? Recombination between homologous alleles starts with a double-stranded cut in one allele, and then, which get with it, I don't really remember this, but then you get resection of the 5' end, so you get this overhanging 3' end. That can do strand invasion, and the homologue basically will then create those holiday junctions. That same process can be used to repair double-stranded breaks, and it can do it incredibly accurately, just as it did the example that I showed. That obviously means that you have to have a homologue. Bacteria are screwed in this way because they don't, right? But for us, if we can use this recombination method, we can repair these double-stranded breaks. But there is another mechanism that double-stranded breaks get repaired by, which I'm almost certainly not going to get to talk about otherwise. But it's called non-homologous end joining. So if there is not a lot of DNA damage, the cell will often just stick together the two pieces of the two free ends of the DNA, which probably is a good bet, right? They may be the only two loose ends of DNA, just stick them together. The problem is that process often leads to small insertions or deletions, which obviously have this problem of changing the reading frame. So the double strand breaks get created, and it's how they get repaired that determines whether they will cause, it'll be a mutation or not. There's one other source, environmental source of mutation. And it's going to come to someone's mind, because many of you have tried to protect yourself against it already.
Sunlight. Sunlight?
Sun.
We just talked
Sun?
about sun.
We just talked about
Oh.
this.
No, we have done the sun, done radiation.
I need a better hint.
We protect ourselves from it.
That's the sun, ultraviolet sun.
Is
Done
it,
with that.
what's it called?
What do you mean by protect? How?
Bro.
How?
Oh, we'll give it away. We'll give it away. It stops
It's
you
like-
thinking.
A lot of things I protect
No,
myself
that's
from.
what you.
Yes, infectious agents, right?
Oh.
Viruses. Viruses. I am hoping that all of you have been vaccinated against HPV.
What was that?
What do we have to?
It's optional, girl.
You vaccinated?
I don't know. Did you take gynecology or
Oh, no,
Right?
I would never.
HPV is a really super common cause of cancer, right? Yeah, I think it's far and away the most common
you
cause.
serious?
Virtually every case of cervical cancer is
It's
caused by
HPV for
HPV,
common cause
right?
cancer? I mean, I'm
This is a
vaccinated.
small virus.
I'm probably vaccinated.
It contains a small DNA genome,
Just
a
one.
double-stranded DNA
Just
genome,
like one of, like a,
which can occasionally
like a one of, to have someone else.
be inserted into the genome.
I hated last.
So it causes large insertion mutations in the genome. It's a big
You
chunk.
want to take two,
Well,
take it.
by all standards, it's not huge, but that is a big chunk of DNA to insert anywhere, right? And obviously, some of these viruses are major causes of common cancers in humans. Those are the environmental things that we might be able to protect ourselves against. But the bad news is there are a whole bunch of intrinsic causes
Wait,
of
so it only
mutation.
adds,
Wait,
it doesn't delete at
I
all?
thought it stops
So-
8,000 pairs, base pairs. It stops.
These are things
8,000
that we
base pairs.
cannot protect ourselves against.
I don't know what the heck actual.
Base.
Number one,
I wanna-
replication error.
It stops 8,000 base pairs.
That's what it says?
Yeah.
What does that mean?
Remember, I talked about this a little bit when I was talking about DNA replication, because I talked about it with regards to bacterial DNA polymerase 3 has a really low
8,000
error
base
rate.
pairs.
It doesn't misincorporate nucleotides more than like 1 in 100,000, 1 in a million times, right? And then what's more, it has the ability to recognize that there was a misincorporation, and it fixes like 99 out of 100 of those errors. But it still leaves occasional mistakes, and as do our DNA polymerases. When we're copying our DNA, which happens with prior to every cell division, there are some mistakes made, and those, if they don't get corrected, go on to be mutations. So that's something that we can't do anything about. Here's one that's really kind of shocking.
So I move from 1 to 2 already?
What is that?
The purine bases are at some quite high frequency spontaneously hydrolyzed away from the ribose, the deoxyribose. That bond to the deoxyribose gets hydrolyzed, and the purine base floats away,
Oh.
leaving us with a site that has no base. There's a sugar and phosphate, yeah, but there's no base at that site, and this is a problem. The shocking part of this has been measured to occur 10,000 times a day
Today.
per
A
cell.
day.
Though this was done in cell culture, I don't think it's a measure that would be easy to get at all from living tissue. So you end up with a site that has, so what was my little? It was TAGG. So this goes to
How was the test?
1, 2,
there it is. My purine's gone missing.
too easy. So if I fail,
When this gets
too
copied,
easy.
Is he really a good
this was good here, of course.
Post videos.
But when this gets copied here,
teacher? Can he explain his questions?
typically an A is just put here, just as a guess. What do you do? There's
Put a
no
T.
base there to inform what nucleotide would be complementary.
Put a-
It's bad. It's bad, and it's common.
What happened? It's bad.
the girl and
Amine groups fall off our nucleotide, off the bases
Oh,
themselves.
so it just falls off.
Now, this is usually recognizable. So cytosine
That's crazy.
cytosine gets deaminated, and it becomes uracil. Now you can see that this is going to be a problem, right? Because cytosine will normally pair with guanine, but if it's changed to uracil, that's going to tell the complementary strand to be an adenine. So it's definitely going to cause a mutation during DNA replication. And this is even worse.
When did we have a U?
Obviously I skipped over the part of the chapter that talks about the control of expression in eukaryotes.
It's okay.
One of the things that we do to control gene expression is to add methyl groups to the DNA. Particularly, we add methyl groups to cytosine. And if you deaminate 5-methylcytosine, you actually get thymine. Which means the cell would have no way of recognizing. We have enzymes that recognize uracil in our DNA. Remove it and replace it with thymine. But if it was 5-methylcytosine that got deaminated, well, it goes straight to thymine. And then the cell has no way of recognizing that there's an error at that particular location. So that's a problem, one that we can't defend ourselves against.
But I thought he said we could add the methyl group.
Our repair systems make errors. The one that I already talked about was this
I'm gonna have to watch it.
non-homologous end joining.
I think he lost in the second half.
It's error prone. And the final thing that we can't defend ourselves against are these things called transposable elements. These are interesting. So these are a large chunk of those interspersed nuclear elements, the repetitive section of our DNA. In their sort of wild form, these are chunks of DNA that are either get cut out of the genome and stuck in somewhere else, sort of a cut and paste function, or some of them get transcribed into an RNA, turned into a DNA, and then reinserted somewhere else, which is sort of a copy and paste function. But who wants these big chunks of junky DNA being stuck into the genome in other places? No one does. It's likely to cause mutations. One of the interesting things about transposable elements is that it is the cause of the wrinkled allele in peas. So, I know I've talked about this a couple of times before. There's an enzyme called starch branching enzyme in peas that enables the buildup of starch into the pea, and without it, there's a lot of glucose get left into the pea, and then it holds a lot of water, and then when the plant dies, the water leaves and the pea becomes wrinkly, right? So if you have the starch branching enzyme, you build lots of starch, the pea stays round. If you do not have working starch branch enzyme, which you're homozygous, then the pea has lots of glucose, lots of water, and when the pea dries out, it'll become all wrinkly. Why does the wrinkled allele not work? Why is it a non-functional allele, a loss of function mutation? It's because some big old transposable element, 800 nucleotides, I think, got stuck into the middle of that gene, which completely throws off the reading frame. So this is an example of a mutation that's due to, and there are lots of others as well, but it's a mutation directly due to one of these transposable elements. Little chunks of DNA that move around the genome. But it's such a bad idea. It's something we don't have to worry about too much. We have whole systems that prevent transposable elements from moving around in the genome. It's another element that I didn't get to cover from that chapter on
You sure you want this pen?
gene expression.
What?
Anyway,
You want this pen back?
they do occasionally move. They've certainly moved in the past. which is why we have so many copies of, I've talked about, when I talked about repetitive DNA, I talked about the alu element, where we have about 500,000 copies of it in our genome. There's no doubt that the movement of that originally caused mutations. Okay, so there are all of these intrinsic causes of mutation that we can't defend against. These are things that are just going to happen. But
Ask him about methyl.
You ask it.
what we really want to do is to be able to defend
You don't
ourselves.
take me seriously now.
And so we have developed a system to be able to identify mutations in the environment, particularly mutations caused by chemicals. Every year in our society, a few thousand brand new chemicals come on the market. You know, biochemists are out there developing chemicals that have new properties that will be useful in some way. Before they reach the market, people want to know, is that brand new chemical that you have, is it a mutagen? Is it going to cause cancer, right? And so there is a whole test that's being developed to try and answer that question. And it's called the Ames test.
Oh, I know
And the
this.
Ames test uses a bacterium, Salmonella typhimurium. I think the reason this is a bacterium that some strains cause typhoid, which is really something no one really wants to get. And salmonella in general causes food poisoning outbreaks, right? But I think the reason it gets used is because one of those bacteria that be incredibly well studied, and so you're able to manipulate it and feel confident that you're not going to lose control of your study organism. So it's actually not the wild type. It's not the wild type. It's kind of special. It's been made more porous to chemicals from the outside than the wild salmonella are, and it has weak DNA repair mechanisms.
Semi-permanent. Think about that. But just not semi.
So it'll be more sensitive to we'll be able to detect mutations more easily. So here's how it goes. Oh, no, the other major factor of it. It is also His minus. And someone's going to guess what that means.
It lacks histidine, lacks the ability.
It's an abbreviation for histidine,
But
and to say that it's His minus means what?
lacks the ability to create histamine or break it down.
Histidine is one of the 20 common amino acids, right?
So it lacks the
When we're
ability to
talking
create
about
it.
amino acids and we're looking at bacterial genotypes,
I don't know.
what does it mean to say that it's His minus?
It can't produce, it can't make it. It lacks the ability to
It
make.
cannot make its own
Bro.
histidine.
Bro.
That's what it means. It cannot make its own histidine.
We hear you.
And so you know how you'd have to grow it in the lab, right? If you wanted to maintain this special strain of bacteria in your
You
lab, you would have to have a growth medium that was supplemented with histidine, because it can't make it for itself. Anyway, so that's my study organism. And I'm going to have a couple of flasks with these His minus bacteria growing in it.
No, thanks.
Oh my god, I'm so stupid.
Don't say that, because I'd be in that group too.
So, we are going to put bacteria into both of these flasks.
Why does he draw flash so well?
So I have cultures of salmonella. into this flask here, I am also going to put the suspected mutagen. You know, whatever that is, the orange color from Cheetos or something like that, right? Something that you're kind of concerned about, right? This flask here is going to serve as our control. One of the really clever pieces of these experiments is that
What does that say? Mutagen?
Mutagen.
they also
That's actually
add
a
some
word.
liver extract into here.
Ew. Jinx.
He or her soda.
And the reason for that is
Yeah, she
there are a whole bunch of chemicals that are known to be okay on their own. But when we ingest them and they get metabolized in the liver, some of the metabolic products, the metabolites of those compounds, are themselves carcinogenic. So the original thing isn't, but it can become a carcinogen in our bodies. And so to mimic that process, the liver extract is put in here. And of course, to make this a really good control, we put that in there as well.
Okay. Might just be having livers everywhere.
And the point of this experiment is to compare these two plates.
Compare.
What should I put in these plates in order to detect the mutation here?
Histidine.
Wait.
What would, if we started with His minus,
Oh.
what would constitute a mutation?
Having his plus.
Sorry?
You would have
His
the histone one.
It's not quite that. What would be, what's the basis of the mutation? Not histone, histidine, but you're right, you're
Oh,
right.
it's histidine.
The mutation that we're looking for is that it goes from His minus to His plus.
No thanks.
And if they've become His plus, what do we have to put in our medium here?
You see this from talking about? It's already plain.
We don't need to supplement at all, right? We could just give them those minimal medium salts and glucose.
That's glucose.
And if this compound that we're testing is a mutagen, what is the expected outcome of an experiment like this? Huh? So we are looking for cells that have mutated from His minus to His plus.
You'd have growth.
If this compound that we're testing causes such a mutation, what would we see on these two Petri plates?
One would have an area for growth of the
Sorry?
histidine. You'd have growth of the histidine, and the other wouldn't.
Yeah, this is exactly it. You'd expect that this plate over here would have a lot of colonies. It's not like this plate's not going to have any, right? There is a background frequency of mutation, right? There is mutation that's going to happen in the absence of any of these environmental factors. Yes?
So,
There's all of these intrinsic reasons that mutations happen. So there will be expected to be some mutants here, even though there was no mutagen added.
you guys.
If the compound, however, that we're testing is mutagenic, you expect to see more mutants on this plate. And you decide that, we can't sell that compound here, we'll sell it in third world countries, right? Oh, I know, that's rude, that's rude. But
Duh.
that's
But
the,
it's
that's,
a
the,
pandemic,
that's the basis of how we solve this test. Is the basis of how we test all these brand new chemicals that come out every single year in our country.
I mean, not historic. Are they historic?
Okay,
These,
I
these are tested.
I can tell. People get antsy. Look, she's racing for the door already. So I will see you on Tuesday next week.
Yeah. Yeah. Does that make sense? No, in class early, so it has history, of course.
Yeah, because they asked me about the
Yeah.
Tell me what he said.
I think this one was actually okay.
I think it was.